the title of the presentation is Redefining the Limits of What's Possible, Sequencing Reimagined. So, and the primary focus of the presentation today is going to be a technical validation of Illumina 5-based technology for DNA methylation EpiSignature detection. So before I get into what the validation was, I'm going to start by introducing the problem first. And the big problem that we have in genetics in general is that rare diseases are actually very common, but very difficult to diagnose. So when we talk about rare diseases, we talk about thousands of conditions that cumulatively affect 1 in 20 humans. Big problem in diagnosing rare diseases is that we have, on one end, very complex etiology. It's not a single mutation, a single gene that causes a disease. It's many types of complex mutations across many different genes and other types of etiologies that cause a phenotype, right? On the other hand, diseases are not defined by a unique phenotype, right? These phenotypes continue to evolve. So if you have complex etiology and you have complex phenotypes, and no wonder we're at the point now where we can sequence entire genomes at very high fidelity, and yet we're plateauing at about 40% diagnostic yield. So the question is the information to what's causing disease in these patients in this remaining genome, and how can we understand it and interpret it? So one possible solution is to be able to identify genetic disease, and I should also say environmental exposures, because we can now also see evidence of epigenetic signatures in environmental disorders and prenatal teratogen exposures via use of the epigenetic fingerprints. So what are these EpiSignatures? So about 12, 13 years ago now, we discovered what is the first evidence of these EpiSignatures, and they're essentially DNA methylation patterns that can be detected in blood of patients, so easily accessible tissue, that are unique for each disorder. Using DNA methylation data in combination with advanced computational techniques that we've developed over the years with EpiSign software, we can now accurately pinpoint specific genetic disorders, not based on the DNA sequence, but specifically based on the DNA methylation profile. So to introduce it further, how do you derive an EpiSignature? You essentially start with a cohort of patients with a common etiology, genetic, environmental, or otherwise, and you generate methylation data. Peripheral blood is the primary source that we use, albeit we're now seeing evidence that it can be seen in other tissues as well, and generating genome-wide methylation data. So historically, and up till this point, majority of this data had been generated both in research and a clinical setting via Illumina EPIC microarray platform. So when we do the genome-wide methylation assessment, we try to look for regions in the genome that are uniquely differential for that particular patient cohort relative to controls. But what's really interesting about this, as we've learned over the years, is that these EpiSignatures in different conditions overlap. So not only do we have to test these patient cohorts against unaffected controls, but we have to continue to test them against other conditions with known EpiSignatures. We use sophisticated methodologies for this, including machine learning, support vector machine, and these EpiSignature biomarkers continue to evolve as we learn more and more about this data. On the right-hand side there, you can see an example of what an output looks like. On the top, you can see a support vector machine output, an MVP score that's generated for growing list of these disorders. This is a patient with a Kabuki syndrome, and they score very high for Kabuki and not for the other conditions, right? And when you look at the clustering of the data, you can basically demonstrate that the patient clusters with patients, not with controls, both by MDS and TSNE and so on. So this is visual representation of what the data looks like. I'm going to mention a few kind of key concepts about EpiSignatures. One, as I mentioned, is the methylation variant pathogenicity score, something we defined over a decade ago. And essentially, it's a support vector machine output score that provides a probability for patient having one of the many EpiSignatures that you can be testing using this particular screen. So these dots, the brown dots in the second column, are individual patients with ATRX syndrome. They score high for ATRX, but they don't score high for other conditions that are in this particular classifier. And then you can see that reproduced for different conditions and for different patients. So it's highly accurate. In other words, highly sensitive and highly specific. It has to be, because when we test patients in clinic, physicians don't have a patient in clinic where they have a suspicion of a patient having a disease. They could have any one of hundreds or thousands of different diseases. So you need to have classifiers that are able to systematically screen for many of these things at the same time. The other concept I want to introduce about episignatures is that while we know that there are gene-specific episignatures, we also know that the genes don't work on their own. They're part of protein complexes, gene pathways. So we know now that there are such things, and protein complex or gene pathway-specific episignatures. I'm showing an example of one. This is buffopathy or Sweet-Sniff remodeling complex. There are a number of different diseases that are caused by mutations in individual genes. They have similar phenotypes, and they also have a common DNA methylation episignature. As you expand the database and you get more and more patients in each subcategory, you can now see subsignatures for individual genes. And what's also interesting is it goes the other way as well. So we also have signatures that are specific below the gene level resolution. So loss of function mutation might have one type of an episignature, gain of function might have an alternate. Particular domain may have a distinct DNA methylation signature, might be associated with a distinct clinical phenotype, and so on and so forth. So you can get a resolution beyond gene level resolution with assessing DNA methylation episignatures for these variants. We've used episignature technology to help define new syndromes. So as we identify new genetic conditions, we find patients with similar clinical phenotypes. They happen to share common genetic etiology. Having evidence, functional evidence of common molecular pathophysiology, having a common episignature goes a long way to trying to ultimately define them as a new episignature, or as a new genetic condition. But what's particularly important for episignatures and clinically applicable is all of you here in the audience know that we struggle in a clinical setting with variants of unknown clinical significance. You sequence the genome, you sequence an exome or a targeted gene, you find that there's a variant that is present in this particular patient. We don't know much about it, it's not in the literature, and so on. If that patient happens to have a distinct episignature associated with that particular gene or that syndrome, this is considered strong functional and/or phenotypic evidence to allow us to reclassify these VUSs. So for a growing number of these conditions, close to 300 now, we have a tool that allows us to reclassify VUSs directly from the DNA source that we use for sequencing. So no need for functional assays, no need for searching other families, no need for co-segregation studies. This is now basically inpatient direct functional evidence of disruption of that particular gene. So this is just an example of a bunch of VUSs in this paper that we published that were easily reclassified to either pathogenic or likely benign via episignature classifier. So as of today, we just launched the first version of this software that is cloud-based end user for clinical laboratories around the world that are doing EpiSign testing clinically. It's called EpiSign Metric. So now labs around the world are able to analyze their own methylation data. Primary use of this is for Illumina methylation arrays, but I'll tell you the rest of the talk about what we're doing next. So essentially, the software allows you to upload the methylation data from array source, and we'll talk about 5-base next. It will provide quality metrics for the patient sample. It will output results. It would flag any potential episignatures with the probability scores that are trained on thousands and thousands of positive and negative controls in the EpiSign knowledge database, which is the largest such database in the world for rare disorders. And it will output a result that clinical labs can now use to issue clinical reports for DNA methylation episignature testing. So a lot of this is really based on a lot of ongoing research. As I mentioned here, there are well over 200, close to 200 institutions at this point, over 30 countries that have been participating in this work. We mapped episignatures, about 300 signatures for more than 200 different diseases at this point. We're actively recruiting patient samples for close to 1,000 additional disorders, so there's kind of a discovery pipeline that's happening in the background, and we're expanding beyond genetic conditions. So we're now seeing ability to detect episignatures for things like fetal alcohol syndrome. So we just published a paper last week in Genetics and Medicine. So a lot of patients that make their way to a neurodevelopmental disorders clinic may not actually have a genetic condition. They might have an exposure to valproate or alcohol, prenatal diabetes, and so on. So we're now able to actually capture the environment effect on the phenotype in addition to the genotype, which is very, very important. And that's never been able to be done before. So with this validation, what do we do? Okay, so the big question is now, as the 5-base technologies are starting to come out, the ideal scenario is if you're able to, while you're sequencing the patient, at the same time generate high-quality methylation data, so you don't necessarily have to do multiple different technologies in order to be able to capture the methylation data. So the first small targeted pilot study that actually presented at the AGBT was built on a small pilot project with Illumina about 2 years ago, where we wanted to see if we can detect patients in this cohort of patients that were unsolved through this program that Illumina had at the time. So essentially, they've done everything on these patients, exomes and genomes and so on. And there's about 20 patients that they sent us for EpiSign analysis to test and see if we can do anything with those patients. And it turned out 3 of them actually had very distinct DNA methylation episignatures. This is based on the methylation microarray data. We went back, it turned out that all 3 of them were found to have specific mutations that ultimately explain their clinical presentation. But what we did for this particular study is we regenerated the data for the same DNA samples, including 17 controls, using 5-base. And we wanted to see if it's going to behave the same as the data that's come out of the array analysis, which is currently the gold standard. And here's an example. So the first case I'm showing you is a patient with Kabuki syndrome. On the left-hand side is the variant that was ultimately determined to be pathogenic based on EpiSign analysis. It was an intronic variant. It was detected through the secondary variant alignment and detection, but it was not prioritized as pathogenic because it was intronic. There was not much known about it. What you're seeing on the right-hand side is the clustering plot of DNA methylation data using 5-base in reference to the microarray data cohorts. So the green cluster are the controls from the microarrays. Blue are controls from the microarrays, or positive controls for Kabuki, and the red dot is the patient for whom 5-base data was generated as a methylation source. So you can see very nice and obvious clustering confirming the DNA methylation signature and pathogenicity of this particular variant. Here's another really interesting example. So this is the second case. Again, this was an interesting case because exome sequencing originally did not detect anything for this case, but episignature analysis, and 5-base confirmed it this time, demonstrated clustering for CHARGE syndrome. CHARGE is caused by CHD1 mutation, CHD7 mutation. And when we looked back at that gene, it turned out that the patient actually had a trinucleotide repeat expansion inside an exon of this gene. This was missed because there was a dropout on the sequencing, and by knowing what gene to look at and having a targeted analysis within that gene, it allowed us to identify this allele expansion and ultimately come up with the cause for this patient's phenotype, and ultimately identifying a gene or variant for the clinical diagnosis. So that was the initial pilot. We got really excited about it because it demonstrated we can generate the data equivalent to the methylation microarray data and with high level of sensitivity and specificity. So we really want to kind of challenge it this time around. So about a couple of months ago, in kind of a hurry, we tried to kind of do a bit more of a complicated technical challenge to Illumina 5-base. So we selected about 45 cases across 22 different episignature conditions. So these are DNA samples that are stored. And we performed Illumina 5-base analysis, and we tested across the EpiSign version 5 classifier that currently screens for about 140 different episignatures. And the data was analyzed in Illumina by Imogen and the Dragon for sequence variation. So a lot of these patients had complex mutations, copy number alterations, indels, deep intronic variants, and so on. And we analyzed the data with EpiSign analysis for DNA methylation. And the result was complete sensitivity specificity. I was actually very surprised because normally what we need to do, we look at an alternate data source. We need to adjust the classifier to compensate for the batch structure and differences. So this is simply plugging in DNA methylation data that came out of a sequencer into an array base, which requires high level of sensitivity specificity classifier to detect. And I'm going to show a few examples here. It's a very cool example. Okay, so this is a patient that is very rare. Type of patient is very rare. This is a patient that has both ATRX and Bekim syndrome. These are 2 rare conditions appearing in the same patient. So rare conditions are very rare. For 2 of them to appear in the same patients is particularly rare. So this is 5-base data, and on the left-hand side, you can see the output of methylation of EpiSign classifier showing the red dot for the one condition, which is ATRX. On the right-hand side, you can see the clustering. So this patient had a mutation in the ATRX gene, which was detected by the sequencing analysis. In addition to that same episignature condition, the patient had an imprinting defect. What you see on the bottom right here, those line plots, patient has hypomethylation in the region for the Beckwith-Wiedemann syndrome. So this patient, using 5-base Illumina 5-base sequencing, we simultaneously detected an episignature for ATRX disorder at the same time as detecting the imprinting defect, which I thought was pretty cool. Another example here that I want to share, and I don't have time to go through all 45 different examples. I just want to kind of selected a few different things that I want to show you. So the other group, I actually selected about 5 patients with the same condition. This is Kleefstra syndrome. And we did methylation analysis. And you can see the output both from the screen of the MVP plots and the clustering and Euclidean clustering down on the bottom here is the very nice data that came out and confirmed the Kleefstra in this particular patient. On the right-hand side, you can see 5 different types of mutations that were picked up by drug and imaging analysis, including some indels, intronic variants, nonsense mutations, and so on. So it completely confirmed the previous findings that we knew, which is we detected a sequence variance and we detect all the epigenetic conditions. But what I want to point you to is the Euclidean clustering, the heat map. You can see down the blue block. The blue block are the microarray-based positive control for this particular syndrome. The green block are microarray-based negatives or normal controls on the other side. And what you're seeing here is the orange are all the rest of the cases in this particular cohort, and the red are the Kleefstra cases. You can see Kleefstra cases very nicely clustering with the positive controls from the array. And you can actually visually inspect the data. It's quite remarkable. Like there's not a lot of variation from this data, considering we only did 45x sequencing, and the arrays are generally equivalent of about 200x sequencing when you look at precision. So I was very, very pleasantly surprised how well it worked. I'll just show you one more example, in this case, Rubinstein-Taby, and I'm only showing you because, yes, again, we're seeing very nice clustering and ability for the classifier to detect this particular epigenetic pattern, but at the same time, I'm showing you here the copy number detection by the Dragon/imaging analysis off of this data. So very nice definitive copy number detection here. So to kind of summarize this kind of analysis and analytical pipeline. So you start basically with, sequencing, 5-base sequencing, and then, once the data's out, you'd go on through the analysis. The analysis essentially is split into 2 kind of components. One is sequence analysis, the other one's methylation analysis. The same data comes out at the same time. When you complete the sequencing analysis, there's a secondary and tertiary analysis, so alignment and variant calling, followed by variant prioritization, interpretation, ultimately at the back end. When we think about methylation, historically, we would have patients that had previously had exomes and or genomes, and then we'd run a methylation microarray, and then we would try to basically interpret the findings that were not there simultaneously. Now we have ability to simultaneously generate methylation data and analyze it. So it gives us really an opportunity by being able to prioritize both secondary and tertiary variant prioritization and calling. So if you have a methylation signature in that data set that says the patient has Kabuki syndrome, you can prioritize your variant calling and variant interpretation algorithms to enrich variants and make sure that we don't miss them. As a matter of fact, vast majority of patients that have exomes and genomes and they're negative, when they get methylation analysis and if they're found to have an episignature, when they go back in the data, we often find the variants. Our tools for secondary and tertiary are not perfect. Anybody that tells you that? I'm not going to say who. They're not lying. They're just being very ambitious. But the bottom line is by being able to prioritize this, and we kind of do this. We do this. HPO terms are one way of doing this, right? You input HPO terms. The problem with HPO terms, phenotypes, is you often end up with dozens to hundreds of genes. And you still end up with lists of variants. Episignatures are pathognomonic. They're highly sensitive and specific. They tell you which gene it is or which couple of genes it is or just which gene family. So that's why I think it's really exciting. The last couple of slides and just kind of overall impressions here. So classically, you do your genomic assessment and you end up with certain level of diagnostic yield. Then you separately may be doing other omics, including epigenomics, DNA methylation, and that adds to the diagnostic yield. Study after study that we've done, both large scale and international studies, we're seeing anywhere between 8 and 15% additive diagnostic yield by methylation analysis, which is quite remarkable. And this is perhaps because a lot of these genes that have episignatures are enriched in these neurodevelopmental disorders patient populations. So you stack these 2 diagnostic yields and you end up with the unresolved. But classically, we had to do multiple different tests to be able to do that. Now we have the ability with high level of precision to do that simultaneously with 5-base sequencing, which is really exciting. The other bit of summary here is basically just to state that we've tested the classifier with a range of different types of episignature conditions, from very robust ones to more mild ones, and we basically in the study kind of threw at it a bunch of different mutational subtypes, and it is very, very exciting to end up with a complete sensitivity and specificity. We are planning to extend the study, for those in the audience, to test a few hundred more patients prospectively and retrospectively in an upcoming study, and I know some of you in the audience already work with us in the space of epigenomic diagnostics. I think that's one kind of key study that we would like to try to complete over the next few months, so you'll probably hear about it in the next little while. But we're really, really excited about this tech. And finally, where do we see the future direction? So I think, of course, episignatures are not just limited to genetic conditions. We know they're related to environmental exposures, the teratogens, lifestyles, behaviors, chronic disorders have them, oncology and hematology, as you may imagine. So having a technique that's able to capture that simultaneously with sequencing will go a long way to being able to interpret the uninterpretable genome at the moment. It's certainly not just for diagnosis. We know that there is now prognostic and treatment response biomarkers related to DNA methylation, the disease severity, and the extent of the robustness of an episignature can be correlated to the clinical and severity of the clinical presentation. And then finally, multi-omic integration. I think by integrating sequence and omics data, DNA methylation being the key low-hanging fruit because it sits on the DNA molecule itself, it's highly robust, it comes from peripheral blood, will be the next main, I'd say, layer of information to allow us to diagnose the undiagnosed portion and learn a lot more.